So I will present our works on open source lab automation here with a subtitle towards applications in precision medicine. So I'm professor in pharmaceutical bioinformatics at Uppsala University in Sweden. And so starting sort of the traditional and much current experimental research is carried out by humans, and it's not uncommon that it's a different person doing analysis. And thirdly, on the right side, you can see maybe a third person being involved in the decision making. So we're trying to advance the use of automation and robotics in the experimental part, automating some parts of the analysis, and also lastly, sort of increasing the use of artificial intelligence in the actual decision-making process. Sort of where do we go from here? And by doing that, we're sort of connecting this into what some people call a data-driven and towards more human in the loop and autonomous systems and sort and alike. But then we see that in some cases we can do complete experiments with robotics, and in some cases we can fully automate the data analysis. And there are even cases where we can automate the decision-making process as well, so we can we'll move towards systems that have more autonomy. So this is sort of a long-term vision that we're going in this direction. So my team builds on 3 pillars: automation, AI machine learning, and cell profiling. And we mainly target applications in drug discovery. So using these pillars to accelerate drug discovery with some applications in predicting safety concerns in drug discovery, elucidating drug mechanisms, screening for new drugs. And as the topic of this presentation is, we're also doing some efforts towards precision medicine. So our workhorse experiment is cell painting, which is basically that we are treating cells with different types of chemical and genetic perturbations, study them using microplates. So we can do a lot of experiments at the same time, stacks of microplates, and then using high content imaging to image these cells in multiple fluorescent dyes. And then we can do traditional image analysis and segment cells and do single cell calculation of morphological features and do some clustering. Or what's been shown to be very successful is also to use these images directly in using deep neural networks in more supervised settings commonly, so that we can make predictions. When we use the cell painting as a profiling technology next to molecular profiling technology, we sometimes refer to it as phenomics. And here we sort of I see this as a technology that you can have in parallel to traditional transcriptomics, proteomics, metabolomics, and similar to transcriptomics, creating profiles of different, for example, differentially expressed genes. We can look at differentially expressed morphology, and then whether these sort of morphological profiles can be quantified with over 1,000 features, looking at different types of properties of the cell's morphology that changes, for example, due to a treatment with a drug. So when we do these types of phenomics, we need to keep in mind that we look at multiple pathways simultaneously, and we don't look at individual protein targets. In fact, we can capture other types of effects. But I like this method because it's relatively fast experiments, and they are very information rich at a low cost per sample. And when doing these experiments, they are by natural untargeted, so we will incubate knowledge over time as we perform more experiments. So here's one example of a small study that we have done, and this study was done more to sort of demonstrate that we could use transfer learning and convolutional neural networks to use these types of images to then make predictions. In this case, it was to predict mode of action for compounds, and so you use compounds that are well known, well annotated compounds, and we see that this type of imaging technology, cell painting, then works well very well in this and many other cases. Here's another study that we recently concluded in my lab. That was for antiviral drug discovery, where we then sacrificed one of the dyes in the cell painting, but added an antibody towards SARS-CoV-2, and we could then, in the same experiment, look at non-infected and infected cells and look at their different changes in morphology when exposed to different types of treatments. And on the right-hand side, you can see an antiviral treatment, and that you, with this treatment, can drive the red dots, which are the untreated infected cells, towards more non-infected cells towards the left. So here's our approach to functional precision medicine, that we are sort of having AI and robotics, and our workhorse, as I said, was morphological profiling experiments. So we want to sort of close the loop between these, working with cell lines and primary cells. So our first take is then to sort of probe these cells using drugs, preferably drugs with well-known mechanisms. And then we can also, based on this, predict and then profile combinations of these types of drugs to these types of cells, and then build up a knowledge base based on this. And then in the sort of applied setting, which I must say we haven't gotten to yet, we're building up for it, is to get primary cells from patients and be able to, with as few experiments as possible, as few iterations as possible, then come up with individualized drug combinations. So this is what we work towards. And as I said, we continue. For every experiment, we build up more data, and I think accumulating enough data here is really the key to success. Drug combinations are challenging, and also, as the previous speaker alluded to, I mean, it's sort of testing all combinations of 3 drugs from a library of 500 is like, it's over 20 million experiments. So obviously we need to have some sort of other type of attack point for this. So can we use iterative experimentation where then artificial intelligence selects the next batch of experiments? That would be better. And if we can use an automated lab to perform these, well, then we would increase the power even more. So, but what would be the objective? What would you optimize towards in these types of selections? Well, I think the more traditional way it would be to select for compounds that selectively kill cancer cells while not harming non-malignant cells. But with the morphological profiling that we are using, we can also try to push cancer cells towards a less aggressive state, for example, in morphological space, and then affecting non-malignant cells as little as possible. So not just always killing them, but pushing them somewhere. If we can't kill them, maybe at least we can make them less aggressive, or move them into a space where we can use some other means to sort of eradicate the cells. So what do we need to achieve this? Well, we need this automated cell painting so that we can carry out these experiments as automated as possible. We need to automate the analysis pipelines. And then we also need this component for intelligent decision making, on how do we proceed with that. So today I will talk mostly about how we automate the cell painting. And I think most of you have seen a lot of news items that appeared during the COVID-19 pandemic about organizations making large investments in AI and automation. So I think it's sort of a lot of focus was placed on this, the speed of drug discovery, and can sort of AI and automation help. And I think it's growing, a new companies that are emerging that are sort of built on the foundation of automation and AI nowadays. And some of them have been really successful in their initial years. But an observation that I make, at least, is that lab automation is relatively rare in academics. And so here we have 3 images from pharmaceutical companies, and that in many cases have more funding available to big, larger installations like these. But the advantages that you have in high throughput and reproducibility, and hopefully also in data quality, it's sort of, there are also some challenges. I mean, it can be expensive, as I said, and you also need quite some expertise in the analysis and data management to be able to use lab automation in your research. And also one observation is that I think this is changing, but traditionally lab automation has been focused on scaling up assays to run the same assay over and over again. And we wanted something different. So this was our, I'm going to tell you a bit about how we're automating our cell-based profiling lab. So we started with the first version 1. This was a fixed setup. It was installed by molecular devices, and it consisted of an image express automated microscope, plate robot, an incubator, liquid handling system, and the proprietary Green Button Go lab automation software. So this was relatively quick to get up and running. I mean, it was installed by then the vendor, in this case Molecular Devices, and they put effort in a fixed protocol. But we also became very dependent on the vendors to solve problems, and we couldn't really sort of expand and service this a lot to us. And that's really what we wanted. We wanted the flexibility to, as academics to expand and work with, integrate open source components with APIs, and also we needed to keep the costs down. So this was not really what we needed. So we sort of set out to instead look at open source lab automation. So we looked around, and we couldn't really find a system available that we could run and adopt and further develop. Either the existing solutions had been gone commercial, or they were not suitable for the way we wanted to build to have a lab automation system. So instead we started building our own on GitHub, and then we procured some instruments off the shelf, a universal robot, UR10E, and then a washer and dispenser from Biotech, and an OpenTrons liquid handling system. And then we tried to integrate them. And then we established some facilities, built these types of microplate cages, and installed these software, and then started building up our own open source solution to control everything. So rely as little as possible on external components that were not under our control. So the first thing was to establish the automated cell painting protocol. So this is the protocol. It's a lot of washers and dispensers, so moving plates around. Start with incubation with exposure, wash, then have a live cell staining, incubate in the incubator, wash a few times, fixation, incubation, washing, permeabilization, and then and in the end you come to the actual staining of the 5 dyes in cell painting, and then incubate and wash and so on. So there are quite a lot of steps to achieve this type of staining, where we get the stains to stain the different organelles of the cells. And so the system then needed to communicate with the incubator, washer, dispenser, and robot arm on the left, and there were different technologies to do that. We had to wrap the software that came with these instruments for example, in the Biotech, that was needed to do a little bit of a bridge with C# to wrap around the DLL files. And for the incubator, well, there was a Java system that we could speak with a TCP socketing and so on. But we've integrated all of these small things, and then wrapped them all in a small REST server, so we can speak HTTP together with them, and then communicate with the different types of system. I think this was pretty smooth to do, but of course it's a little bit of reverse engineering in some cases, depending on the systems that you're investigating. And then in the middle, comes one thing that we did not expect from the beginning. So the robot comes with a GUI to program, but we actually didn't really find that sufficient. So we created our own GUI for creating the robot arm moves so that we can very, very precisely, using the keyboard, move 1 millimeter left, right, and so on. So to control to guide exactly how the robot arm should move and where the different positions. And then the scheduling of the protocol so that we can run multiple plates at the same time. For that we used constraint solving and optimization. Don, data engineer in my team, made a mini programming language, a DSL, where that allowed for easily expressing the protocol in code so that it could be scheduled. So it's relatively readable, and then you could execute this in different ways for live simulation and symbolic. That was sort of the different ways so that we can test them both in a simulated environments and then run them live in that sense. And for the optimization, he used an SMT solver to then to find an optimal scheduling between the different plates. On the robot arm, we put a camera, and Rickard here in the team then made a machine vision system, and we primarily use it for barcodes right now. It's prepared to do more quality control steps. Is the plate there that I'm going to pick up? But for now, we mainly use it for continuously looking at barcodes if we're picking up something. And then we have within our system, we also collected a lot of sensors, and we can have control of temperature, humidity, and as such that might affect the different experiments. So here's an example. You can see Dawn here. I'm going to fast forward a little bit onto this. Dawn has been working with the robot. Here you see a lidding and delidding. So you have 3 cameras. They're all relatively in sync. They're just in 3 different positions, and we see the robot arm moving it now into the incubator, and now issuing a rest call to the incubator, pick up the plate and incubate it for some time. And the system then comes out and takes over and continues with it. Well, it's just a taste of the system here. So the throughput of this, it's very, very then parallelized, so that we can run 12 plates in parallel, 12 microplates, and it takes about 2 hours to do the actual cell painting. The first step of 24 hours is just waiting in incubation for exposure time, but that's not the interesting part here. And if you look at these 12 plates, if you then see how much information you get from them, let's just say we don't use the outer wells because of edge effects. That leaves us with 308 wells. We have 5 fluorescent channels, and let's say we take 3 bright field Z planes, and then we do 6 sites within each well. That leaves us with over 177 images, or 1.4 terabytes. So we really get a lot of data coming out of these experiments. So microscopy imaging quickly becomes the bottleneck. Our microscope cannot keep up with this, obviously. So that's something that we would like to remedy, of course, in the future. But microscopes are expensive, so we actually have an ongoing project to build more microscopes and to build these open source microscopes together with our 3D printing facility, and it's looking really promising. And we'll get back to more when we have more information around that. But obviously we're dealing with large-scale data here. So we actually have spent a lot of time in the parallel while building up the hardware, also building up the infrastructure and the data pipelines to work with these. So we've installed some on-prem hardware. We have CPU servers, GPU clusters, and storage, and so on. And we really work with virtualization and the microservices and the Kubernetes environment to be able to have a nice fault-tolerant environment to do it. I'm not going to talk more about this today. I'm just going to give some highlights of some other open source tools that we've developed to aid us in this. So one is the imaging database that Anders in our team have done, and the web viewer, because the first thing people want to do during an experiment, you want to see images coming in and inspect them and so on. For going from microscopy imaging to segmenting cells and then calculating these morphological profiles I talked about earlier. We use the Cell Profiler software from Broad Institute, and we have also automated these analysis pipelines on our Kubernetes cluster so that we can run different pipelines, for example, for different cell types that we have, and we'll keep track of the analysis that are running. And then in the same system, I mean, this is all within the same Kubernetes clusters, and the same. So we just open up notebooks, and we have access to the data that has been produced, both in terms of images and both in terms of the calculated profiles. And then in these notebooks, we can do the QC and the data analysis, the downstream analysis that we do. So that's I think it's a system that that is starting to become extremely powerful for us. Now, we haven't really gotten to this part where we continuously updating the AI models, but I've been working on the parallel projects on this problem for quite some time, and it's also formed a spin-off company, Scale Systems, that really works on both federated learning and ML ops, the system for how can we continuously, if we have new data coming in, can we continuously update models, version them, and put them in production with proper monitoring and logging, etc. So this is something that we're getting to the point of using, and that's we have the system already in place. So this is my final slide. Some perspectives on open source lab automation. There are some really clear advantages for us. We have control of all components. If it breaks, we can fix it. If we want to expand it with new things, we can do it. We built all our infrastructure as code, so that is perfectly portable, and we can deploy it on our local servers or on any type of cloud environment. The lab automation as code as well. I mean, we have full control of it, so we can. If we find something that doesn't work, we can fix it. Obviously, it takes time to develop it the first time, so that's an overhead. The analysis pipelines is code, and we also 3D print the missing pieces. And I think putting all of these components as open source, I think that is. We hope that will be useful for others as well. As I said, we have full control of the scheduling, so that means we're prepared for this intelligent decision making that we're working towards. We're trying to realize in our lab. There are some downsides of this, of course. It takes more time to set up these and build these systems than to buy a ready-made solution. You also require to have competence on using hardware, software, and data engineering expertise within your lab team. I would say, and with discussion with different companies and pharmaceutical companies, that it's probably more suited for academic labs in this way. And I'd like to sort of conclude with saying our system, which we call AROS, we built it for ourselves. We didn't build it for anyone else. We're very happy if other people use it. Even happier for collaborators and the pull request. That would be fantastic. But it is something that it's work in progress. But I'm expecting this to be work in projects for many years to come, and it will continuously be improved. And in the lower right corner, you have the link to this. And we're working on our first manuscript to publish this. So the documentation is probably lacking quite a lot now, but it's something that will become a lot better in the coming months as we are moving further towards sort of doing some more outreach on this. So with that, I thank you for your attention. Looking forward to questions. If you have
Wonderful. Ola, thank you so much for your talk. I have to say that as you were speaking about writing code and debugging code to move a robotic arm, I was wondering, how many casualties have you had in the lab as a result of various bugs?
But I mean, obviously there are things that happens that you did not intend to, and that I mean, we sort of the robot works. I mean, it's contained in its own cage, so
I see.
No animals or humans have been harmed during the development of this system. But
That's great to know.
there have been some accidents that has sort of broken some parts. That has
I imagine the same wouldn't be true of the material losses.
Yeah,
So
they have not been large, but I mean, to some extent, you have to accept that it's a development and some things might happen. We have not had any really expensive accidents yet, so it's sort of
I see.
more on the level of that. During development, you drop a plate here and there and so on, or you
smash
there
a
has
sample.
been some, yeah, this sort of you crashed into the robot crashed into the Plate Hotel, for example, which is sort of.
I see. Makes sense. My kind of more serious question was really just about scaling this. So you mentioned that this is all open source. You started with something that was not so much open source, and you can really modify and tweak it to make it work for you. But I suppose obviously the kind of places which would be really excited about this sort of advancement is really big pharma. If you've got a research program where you're focusing on an indication and you need to screen millions of drugs, that it appears like something like this would potentially be quite useful. So but obviously approaching big pharma and actually creating any sort of partnerships that can take potentially years in the making just to close the deal. Have you had any experiences talking with pharmaceutical companies and seeing if there would be any interest, if they would be interested in sort of rolling it out at the sort of bigger scale?
So it's a very good question, and it's something. So first, I think the tradition in pharma is usually that they rely on external vendors to provide them with the complete systems. So they don't really have in-house expertise in developing these systems, and they buy complete solutions. I think that is the more traditional way for big pharma to work with the laboratory automation, and that they sort of come install entire systems, and then they take responsibility that these systems run and they fix them if they break and so on. I think we will see a change in the future. But I think that these vendors offering these services, they will be pushed into the way that from scaling up individual protocols to be a bit more wide in the repertoire what they offer. But I also think that I'm hoping that maybe some companies would be interested in these kind of approaches. At present, I think it would take more time, and I also wrote I think it's more suitable for academic labs right now. Who knows? In a few years, maybe things have advanced further and so on. But with that said, I've also had discussions with more startup companies who are starting from scratch.
Exactly. Yeah. That's what I was going to say.
You could have our automation from scratch. How should we do it? I mean, it's sort of, and I'll try to share what the experiences we have, and I would be extremely happy if there are some other companies or academic groups that are interested in sort of building up something that would sort of reach out to us, and we could work together. I mean, that's the power of open source. If others can do it, you can sort of join in.
Yeah, actually, one of my friends was an early investor in Synthase, which is a lab automation company based in the UK. So I'll definitely follow up and see if we can make some introductions. So that we can raise awareness of your project. Ola, we've got one more question that's actually from the audience. So I'll just bring it up on the stage. It's specifically the question is specifically about the domain-specific language that was used to control the robot. So the question is, have you tried to use something that's already established as opposed to writing your own?
Yeah, and I think it is a good question. And we looked around and we tried. We have looked at Anta, for example, and it's I think it's from our perspective, it has been pragmatism that we found it a bit difficult to sort of to install and use and configure Anta in that sense. So and doing our own DSL. I mean, it sounds like we're inventing something. I mean, Anta is a solution that sort of that can be used by others. I mean, it's a tool that's facing outwards, and we wanted to solve our problem only. That we did our own DSL. That was just because it was convenient. So it's not a general solution that can be used by everyone. It's something we did to solve a problem with us. I think that's my short answer to this question.
Yeah, wonderful. That's really good to understand. Ilya, thank you so much for asking the question. Ola, I think it is time for us to be moving on to the next speaker. So yeah, thank you so much for a great talk. And I'm looking forward to hosting you perhaps in one of the future events.
Thank you very much.